We have now seen what is meant by a monohybrid cross. It is a cross involving one pair of contrasting characters. Similarly, a dihybrid cross, it is a cross involving two pairs of contrasting characters. Monohybrid cross is in accordance with law of dominance and law of segregation. So, law of dominance and law of segregation both are applicable to a monohybrid cross. For a dihybrid cross, law of dominance and law of independent assortment. So, we have seen all these with suitable examples. In the same way, there can be a trihybrid cross, a cross involving three pairs of contrasting characters. A tetrahybrid cross, a cross involving four pairs of contrasting characters. Now, let me just give you some idea about a trihybrid cross and how to get the combination of gametes for a trihybrid cross. So, a trihybrid cross is a cross involving three pairs of contrasting characters. Let us represent the factors or genes responsible for these three pairs of contrasting characters as capital A small a, dominant recessive, capital B small b, again dominant recessive, capital C small c, dominant recessive. When you have only one pair of contrasting characters in the heterozygous condition, you get only two types of gametes. When you have two pairs of contrasting characters in the heterozygous condition, you get four types of gametes. When you have three pairs of contrasting characters in the heterozygous condition like this, you should get 8 types of gametes. 8 types of gametes. How to find out that? You can write a simple method. One pair of contrasting characters represented by capital A, small a. For this, capital B, small b, and again capital B, small b, and for this capital C, small c, capital C, small c, capital C, small c, capital C, small c. So, these three, this will be one type of gamete. Capital A, capital B, capital C, A, B and small c. A, B, small c, another type of gamete. A, small b, capital C, another type of gamete. A, small b, small c, one more type of gamete. Then small a, capital B, capital C, another type of gamete. Small a, capital B, small c, another type of gamete. Small a, small b, capital C, one more type of gamete, and small a, small b, small c. So 8 type of gametes are possible. These are the possible gametes. Mendel conducted experiments involving trihybrid cross also. In addition to all this, Mendel has made one more significant contribution, and that significant contribution is known as test cross. Or back cross. A test cross or a back cross is a method of verification devised by Mendel. It is a cross between heterozygous, it's a cross between heterozygous plant and homozygous recessive plant. Let me explain this with an example. In tall plants, we have seen there are two possibilities. There can be a homozygous tall plant, or there can be a heterozygous tall plant. For external appearance, both of them look similar. Phenotype is similar. Phenotype is tall, but genotype is different. Suppose you come across a situation, you are given a tall plant and you are asked to find out whether this tall plant is homozygous or heterozygous. You can do it by conducting a test cross. So I will say, take the unknown plant, conduct a cross pollination with homozygous recessive plant. So you have been given a tall plant, you will be crossing it with homozygous recessive plant, that is dwarf. When a tall plant is crossed with a dwarf plant, in the next generation, if you get all tall plants, it is a homozygous plant. If you get tall and dwarf plants in the ratio 1:1, it is a heterozygous tall plant. So how is it? It is like this. Tall plant can be either capital T or capital T small t, whereas dwarf only small t. Okay, so here the gamete is capital T, here the gametes are 50% capital T, 50% small t. Here all gametes are small t. Suppose these two gametes meet, tall and dwarf. The result will be capital T. So all the resulting plants will be tall. On the other hand, if it is a heterozygous plant, see what happens. This and this will give you capital T, small t. This and this will give you small t, small t. So you are getting tall and dwarf plants in the ratio 1:1. So this is known as test cross ratio. So this is a test cross ratio 1:1 for a monohybrid cross. Similarly, for a dihybrid cross, you can conduct a test cross. For example, if you take the roundello plant of the F1 generation, the roundello plant is heterozygous, but we don't know whether it is homozygous or heterozygous. So it can be capital R, capital R, capital Y, or capital R, capital Y, small y. This is crossed with a homozygous recessive plant, small r, small y. Here the gametes will be capital R, capital Y. Here the gametes will be capital R, capital Y, or small r, small y, 50% each. Here the gamete will be small r, small y. This and this should give you all roundello plants. But if it is heterozygous, this and this, this and this, so you will get capital R, small r, capital Y, small y, small r, capital Y, small y. Again you are getting dominant and recessive 1:1 ratio. So the genotype of an unknown plant can be ascertained by conducting a test cross. Sometimes it is also called as back cross. Suppose it is crossed with a homozygous recessive plant, small r, small y. This produces only one type of gamete, capital R, capital Y. But here this is heterozygous. We have seen it produces four types of gametes: capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. This produces only one type of gamete. If these two fuse, it will be capital R, capital Y, roundello, only roundello. No other plants will be produced. But here, if these two fuse, capital R, capital Y, small y, this will be a roundello plant. If these two fuse, capital R, small y, it will be a round green plant. If these two fuse, small r, capital Y, small y, small r, capital Y, small y, it will be a wrinkled yellow plant. Small r, small y, wrinkled green plant. So for the same four combinations, you will get a ratio of 1:1:1:1. This is the test cross ratio for a dihybrid cross. This is the test cross ratio for a monohybrid cross.